So now I'm exciting the spinal cord with green light and seeing red light being emitted from certain neurons. This particular fish has a red emitting dye in all of the excitatory neurons of the spinal cord.
Students at the University of Minnesota Neurophysiology Lab are using zebrafish to unlock some of the secrets of human biology.
So I'm a second-year graduate student in the neuroscience program, and I'm interested in the mechanisms that underlie vertebrate walking, swimming, running, just locomotion in general. So I'm using a combination of genetic techniques and electrophysiology to really try to pin down the details of how all that works.
The focus of this lab is on studying neural circuits that are intrinsically rhythmic that drive locomotor behaviors. And we use zebrafish for this. So for humans, the way we walk, we basically alternate our muscle contractions of our legs, left to right, left to right. And dogs, cats, mice, rats are similar. The way they swim, they have these lateral undulations. We call it lateral undulatory swimming. But really the way they swim is they kind of do a little shimmy in the water.
They are actually a freshwater fish from India originally. As an adult, they're probably about 3 inches long, but we actually study them at about the 4-day stage. These are 4-day-old larval zebrafish. They have an air bladder, so they can swim up in the water column. They can also forage for food, and so this is the first day that they actually go and hunt for their own food.
Central to everything at the station is the fish. We have 2 electrode holders that hold very fine glass pipettes with tips that range between maybe 1 micron and 20 microns. And with those pipettes, you can do things like attach to a single cell and record its electrical activity. We have to take those tiny electrical signals from the electrodes and feed them into an amplifier. And the amplifier takes those signals and in a very clean way magnifies them to the point where we can see them. Those then go through an analog digital converter and then get fed into our computer system where we can acquire data and analyze it. And then I guess it comes to me.
The students aren't just collecting data. They hope to one day apply their research to people with locomotor disorders.
This is way down in the future. We're at the very, very cellular basis of this, but different locomotor disorders, Parkinson's, I believe Huntington's, or even paralysis. When people have spinal cord injuries, what can we do? What is the circuitry and what has broken in that circuitry for people with these different disorders?